Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the origin of life on the earth. And in this context, we have discussed about the various theories. We have also discussed what was the condition of the earth so that it was possible to have the life on the earth. We have discussed that what is the structure of the earth and so on. So in the previous lecture we have emphasized more on the conditions and the the way in which the life was originated onto the earth. And in that context we have discussed many types of theories. We have discussed about the theory of spontaneous generations, we have discussed about the theory of special creations, then we have discussed about the theory of the abiogenesis and so on. By discussing those theories, we have understood that these theories were mostly being based on the different types of experimental different types of assumptions. So they were not based on the experiments, they were only based on the assumptions. For example, in the theory of special creations, this is a completely a religious theory where the people have discovered or people have believed that God is the creator of the earth and the different types of organisms, and the different religions have their own way of explaining this particular phenomena. And then we have discussed about the theory of spontaneous generations, where the people have said that the non-living matter give rise to the different types of organisms. And then subsequently, so there were six major theories what we have said in the previous lecture that we are being proposed. Out of these six theories, the five theories were discussed in our previous lectures, and all these theories were mostly being based on the assumption as well as without performing the much of the conclusive experiments. So now in today's lecture we are going to start discussing about the modern theory or the chemical theory of the origin of life, and that explains that how the different chemical molecules give rise to the generation of the organisms. And this theory is, apart from the earlier five theories, this theory was completely being dependent on the different types of experiments which are being performed, and it is based on the scientific evidences. So let's start discussing about the modern theory or the chemical theory of the origin of life. So what we have discussed about the five different theories. We have discussed about the theory of special creations, we have discussed about the theory of spontaneous generations, we discussed about the theory of cataclysms, we discussed about the theory of cosmozoics, and we discussed about the theory of eternity of life. Now in today's lecture we discuss about the modern theory. So what is the modern theory? The modern theory, which is also known as the chemical theory or the theory of the primary abiogenesis. So modern theory is very much linked to the theory of abiogenesis, except with the condition that what they have said is that it is not possible to have the life on the earth under the current earth conditions. So we have to create the conditions what was present on the primitive earth. So if you recall, in the previous lecture we discussed about what was the condition on the primitive earth, right? The condition on the primitive earth was that it was reducing in nature because there was no molecular oxygen was present, and it was mostly the hydrogen and the hydrogen compound, right? Whether it is the water or the hydrogen or the methane or the ammonia, all these molecules were the hydrogen derivatives. So these molecules were making the environment more reducing, and we have discussed in the previous lecture as well that why the reducing environment is good and what was supporting the origin of the life. Although the life cannot continue without the oxygen because it depends on the various processes which requires the energy, and that energy is being released only by the oxidation process. But earlier in the primitive earth conditions there was no dearth of energy, because the energy, they were getting the energy from the solar energy, they were getting the energy through the the volcanic eruptions. They were getting the energy through the lightning and all other kinds of sources. They were getting the energy even from the radioactive decays and so on. So the conditions in the primitive earth was the condition on the primitive earth is different from the present condition, and which do not permit the abiogenesis. So the people were trying to do the abiogenesis. If you recall the classical experiments which are done by the Reddy's or Spelagini or even the Louis Pasteur, they have disproved the theory of abiogenesis simply because the current condition of the earth is not supporting the life. It is not supporting the origin of the life. It is only supporting the life activities because it is supporting the generation of the energy, but it does not permit the abiogenesis. The idea of the chemical theory was put forward by the two scientists, A.I. O'Farin and the J.B.S. Haldane, and it has made the following assumptions. What are these assumptions? The spontaneous generation of life under the present environment is not possible, and that's why that is the reason that the abiogenesis theory was disproved or which was discarded because of the classical experiments which were done by the Reddy and Spallanzani or the Louis Pasteur's. And the Earth's atmosphere approximately 1 billion year is very different from the current condition. So the Earth atmosphere what was there approximately at the 1 billion year is very different because current condition is more oxidizing, right, whereas at the time of the 1 billion year when the earth formed, it was mostly be reducing, right. So it has more of the hydrogen and the hydrogen related molecules. The primitive earth atmosphere was reducing in nature, and that was very, very important. And under these conditions, the chemical molecules like the inorganic molecules like the water, ammonia or the methane, they were reacting with each other and through a series of reaction to form the organic substances and the other complex biomolecule. The solar energy and the UV radiation provide the energy for the chemical reactions. Now, when these two great scientists have proposed the modern theory of the origin or the chemical theory of origin, they were also so scientists have said, okay, this is fine, but now how you say that this is actually be the case, right? So you have to perform the conclusive experiment. You have to show me that this is actually be the true. Then for that experiment, for that reason, the scientists have started doing the experiment to prove or to disprove the modern theory. So the first scientist who did the experimental evidences to prove the theory, modern theory, because initially the hypothesis which was proposed by the Haldane did not get much scientific attention or the support because there was no experiment which are being done by the scientists. And then to conclusively support the chemical theory, the Stanley Miller and the Urey conducted the experiment mimicking the primitive earth conditions. So how they have done that? They have actually developed a scientific experimental setup which contains a glass flask, a condenser and a liquid flask, and which are interconnected with a tube with the source of electric spark to provide the energy. So what they have done is they have taken a experimental setup, and where they have having the the glass flask, and they will be connected to the liquid flask, right? So this is a liquid flask, right, where the water can be boiled, and then they have also have the glass flask where they are actually been able to produce the electric spark, and that electric spark is actually mimicking the lightning, and that's how they were actually been providing the energy. And then they can actually be able to allow the entry of the different types of gases into this. So what they have done is they have taken the methane, ammonia and hydrogen as a gas into this chamber, and then they were having the condenser. So that condenser is actually going to cool down these gases, so they will be present in the liquefied form, and or the substances whatever is going to be formed. So then what they have done, they have taken this apparatus, and then he introduced a mixture of methane, ammonia and hydrogen in the ratio of 2:2:1, and the water and water vapor at a temperature of 800 degrees Celsius. If you recall, when we were discussing about the earth formation, we said that earth is been formed when the temperature was approximately around 1000 degrees Celsius. So they have maintained the similar kind of primitive earth conditions like 800 degrees Celsius. They have mixed the they have made the environment reducing by introducing these hydrogen in the environment as well as ammonia and methane. So there was no oxygen, so no oxygen in their apparatus. Then they have, what they have done, they have actually circulated this mixture into this apparatus. So this is a closed apparatus, right? There is no way you can actually get it can go out, right? So if, because the outside is oxygen, right? So if oxygen will enter, it will actually going to destroy the chemical reaction, and that's why there will be no generation of the organic molecules. Then they allowed to circulate the mixture into this closed glass apparatus for 18 days continuously. So they have allowed this to be circulated. So the they have heated the water, right? So water is going this way, right? And while it is going, it is also taking the different gases, right? Different gases into this mixture, and then these gases are entering into this bulb, right? And once they enter into the bulb, they are actually putting the electric shock. So because of that, there will be electric spark, and that actually mimics the lightning, right? And that's how it is actually providing the heat or the energy into the system. So because of this energy, these gases are actually going to react with each other, and then they were actually going to be form the new compound, and that's how they will be get cooled down by a condenser. So they will be form the liquid, and then these gases are actually going to the liquid is going to be condensed in this particular trap, and after that the gases will again enter into this water. And once they enter into this water, they are actually going to come out in the form of a kind of a volcanic eruptions or something, right? So they will come out from this water, and again they will travel. So this continued for 18 days, and during these 18 days they have taken out the sample. So they have a sample port, what you see here, right? They have sample port. So whatever the liquid condense here, they can be able to take out the sample, and then that sample can be tested for the synthesis of the new compound or not. So they provided the energy in the form of spark by supplying electricity at the 75,000 volt to the two electrodes. The electric sparks mimicking the lightning what is happening in the primitive earth atmosphere. While passing the mixture, gases were passed through a liquid flask to stimulate the volcano. The mixture was collected from the stopcock and analyzed using the chromatography as well as the calorimetric techniques. So chromatography is a technique, or the calorimetry is a technique. So calorimetry is actually going to tell you what will be the energy level, or what was the the free energy change of these compound what are being formed, whereas the chromatography is actually going to tell you the nature of that particular compound, whether the glucose is being formed, or whether the sugars are being formed, or whether the organic molecules are being formed or not. So chromatography is a separation technique which actually not only going to separate the molecule, but it also going to tell you the nature of that particular compound. The chromatography can be also be used to to purify or to characterize the molecules. The analysis of the mixture indicate that the presence of amino acids such as glycine, alanine, aspartic acid, nitrogenous base like adenine are being formed into these particular experiments. So this proves that you can be able to by doing this experiment you can be able to synthesize the new compounds. So the mixture what they have collected from the stopcock, analyzed using chromatography and calorimetric method. The mixture of this indicate the presence of amino acids such as the glycine, alanine, aspartic acid and nitrogenous bases such as adenine and the simple sugar ribose. So you know that the adenine and plus sugar is actually can give you the simple molecule which is called DNA or nucleotide. So these two molecules can actually will be able to give you the DNA as well as the RNA, whereas all these molecules like glycine, alanine, aspartic acid and are probably will be good to give you the proteins. So that's how they have proved that by doing this simple experiment, and where they have not done the experiment for a very extensive period, like 18 days is a very small period, but to see that the protein are formed or not, right. So 18 days is good enough to say that by doing this method you can be able to synthesize the biomolecule. So once you synthesize the biomolecule, they can be able to give rise to life. So the chemical molecules, the chemical reaction which explains the synthesis of these compounds are as follows. The formation of the small simpler organic molecule like the formaldehyde or the HCN. So the carbon dioxide is actually going to be dissociated into the carbon monoxide and molecular oxygen, and then the methane is actually going to react with these two compounds to give you the HCHO, right. This is the formaldehyde, and then the water is going to be released. Similarly, the carbon monoxide which is going to be react with ammonia is actually going to give you the HCN, and if the methane is also going to react with the ammonia, it also can give you the HCN. Similarly, you can also explain the formation of the glycine. The formaldehyde, ammonia and HCN then react to form the glycine. So this is the formaldehyde, then it reacts with HCN and then it reacts with the ammonia, and that's how it is actually going to give rise to the glycine. And then this NH2CH2CN is actually going to be react with water, and that's how you are actually going to have a molecule of ammonia, and you are also going to have the molecule of glycine. So this glycine, the generation of glycine and the generation of the other biomolecule proved for the first time that it is possible to have the synthesis of the biomolecule by the inorganic molecules. Before that, it was not possible, right. So because the condition in which the people were trying to prove the abiogenesis was not conclusive. Like for example, even if you take the rotten meat, it will not give rise to the the molecules. So this classical experiment of Stanley-Miller actually proved that the modern theory or the chemical theory is correct, and it actually can be able to give us the insight how the life is originated onto the earth. Then they move ahead and they have actually given the detail what are the different steps could have happened by synthesis of these biomolecules, and then how these biomolecules could have given rise to the primitive cell, and that primitive cell could have been evolved into a more and more complex organisms. So according to the chemical theory, so what is the proposed events, right? So nobody knows how the life is originated onto earth. So but these scientists, based on these experiments, they have proposed the multiple events. So what you have is, you have the 4 major steps what they have proposed. So according to the chemical theory of origin of life, a series of chemical synthesis give rise to the life, right? You have seen how the carbon dioxide and ammonia and the methane is actually reacting to give rise to the formaldehyde, and then formaldehyde is reacting with these molecules to give rise to the glycine. So synthesis of glycine is the simplest amino acid, but then once the glycine is formed, you can also have the multiple different combinations, and that's how you can be able to have the synthesis of the even complex biomolecule. So considering these scientists have proposed the events which probably could have happened in the primitive earth environment, and that's how they have said that it is a 4 major step process. What is the step 1? Step 1 is the formation of inorganic molecule. The high temperature of the primitive earth did not allow the condensation of the atom to form the inorganic molecule. As the temperature of earth goes down, the condensation of the different atom molecule give rise to the simpler molecule. For example, in the primitive earth, you have the hydrogen, you were having the carbon, you have nitrogen and oxygen. But when the temperature goes down, these molecules were coming together, and that's how they have synthesized the simpler inorganic molecule like ammonia or the acetic acid. So you can see that the oxygen which was molecular oxygen which was associated with this molecule, and that's how it has given the two simpler molecule which are ammonia and amino acid, right. Apart from that, you can also have the methane, and the elements, the most abundant element are the hydrogen, oxygen, nitrogen and carbon. The reaction of these molecules give rise to the different gases such as hydrogen, nitrogen, ammonia, methane, carbon dioxide and water vapor. So, because these elements were present, you can have the different types of gases. You can have the nitrogen, you can have hydrogen, you can have nitrogen, you can have the ammonia, right. You can have the ammonia, you can have the carbon dioxide and you can have the water. And water, because the temperature is high, it is always going to be present in the form of vapor. So water is actually going to react with will be a medium through which these different gases are actually going to react with each other. The energy of these reactions was proved or was provided by the sunlight, lightning or the volcanic eruption. Because if you want to do a chemical synthesis, you have to form the bond, right. For example, you have two carbon molecules, then if they will not form the bond, right, they will compound, they are supposed to form a bond, right. They are supposed to form a bond, like whether it is a single bond, double bond or triple bond. This bond formation requires the energy. So that energy is going to be supplied externally, and that is being supplied by the sunlight, lightning or the volcanic eruptions. So in the step 1, you are going to have the formation of inorganic molecules, like inorganic molecule, whether they are the gases or the liquids. Then in the step 2, you are going to have the spontaneous formation of the monomeric organic compound. The simple molecules interact with each other to form the simpler monomeric organic compounds. So you can imagine that you have the primitive ocean, right, primitive ocean, where you have all sort of gases like methane, ammonia, carbon dioxide, hydrogen, nitric oxide and all those kind of molecules. So when they will, and then you have the lightning, right, and you also have the UV light from the sun, and all these are, when they are actually forming the simpler monomeric compounds like fat, proteins, nucleic acids, primitive water and the polysaccharides. So these molecules are like sugar, fatty acid, glycerol, amino acids and the organic bases, like the organic bases which are purine or the pyrimidine. The reactions between the inorganic to give simpler organic compound occur in the reducing environment inside the ocean. The inorganic molecules were condensed in the form of rain as a temperature of earth goes down. Hence both inorganic compound and the simpler organic compound were present in the primordial ocean, which means once the temperature goes down, the water was condensing, right. So there was a rain, right, and because of the rain, all these molecules what are present in the atmosphere are condensing and getting dissolved into the molecular ocean, and that's how they were getting a medium in which all these molecules are reacting with each other. So energy they are getting from the lightning as well as the sunlight, and that's how they are forming the simpler sugars. They are forming the simple fats and simple nucleic acid molecules like the nucleotides. And all these molecules are present in the primordial oceans. Then in the step 3, the spontaneous formation of the complex organic molecules. So since once you have generated the simpler organic molecules, these simpler organic molecules are actually going to react with each other. So what we have generated the simpler carbohydrate like simpler sugars, and these simpler sugars are then actually going to form the more and more complex sugars. So the small simpler organic compounds react to form the complex organic compound. The simpler organic simple amino acid reacts to form the polypeptides. Sugar reacts to form the large sugar molecules. Fatty acid and glycerols combine together to give you the fat, and the heat of the sun is utilized for providing energy for these reactions. So what we have is you have the simplest organic molecules. They will react with each other to give the smaller, more complex like molecules like the glycerine, ribose, glucose, and on even more larger molecules. So fat is also being developed. Once you have the glycerol and the fatty acids, the glycerol and fatty acids are actually going to combine to give you the fat. And you know that the fat is a very important component of the plasma membrane, right? So that is actually going to give rise to the plasma membrane of the primitive cell. So that we are going to discuss. So in the step 3, you are going to have the synthesis of the complex organic compounds. Then in the step 4, you are going to see the generation of the smaller molecular See, once you have generated the small molecules, they are actually not going to have the tendency to club together, because they can have the, but once you generated a bigger biomolecules like the proteins or the nucleic acids, they will be actually going to react with each other, and that's how they will be actually going to form the complex, right? So they will be going to form a complex, and these complexes are known as the coiservates, right? So they will be formed as they will be called as the coiservates. And you can see, right? So all these molecules, so where you can probably there will be a protein which is actually going to form outside layer, and then within this you can have the DNA or lipids and all those kind of things, and that's why you are actually going to have a ball shaped coiservates where the molecules are going to come together and they will form the colloidal aggregates which are called as the coiservates. A layer of water molecule forms around the protein molecules present in the coiservates. So once the protein molecules are there, and you know that and even if we are going to discuss in future that protein molecules are actually being made up of the amino acids, and amino acids are going to attract the water. So what will happen is the water molecule is actually going to absorb onto these molecules, and that's how you have a coiservates. And apart from the coiservate, outside the coiservates, you are going to have the plasma, the layer of water. And because of this small area is actually going to be turned into a very, concentrate. So it will actually going to concentrate the reactants, and that's how it is actually going to facilitate the chemical reaction at a faster rate. You know that the chemical reactions are dependent on the concentration of the molecules, right? So if A is getting converted into B, if you have the 10 milligrams of A, it is actually going to be form the 10 milligrams of B, right? But if you increase the amount, right, if you increase the, even if you keep the 10 milligrams and the concentration is 0.5 milligrams per ml, the reactions are actually going to give you the reactants in the range, in the same ratio. But if you take the 10 milligrams and make the concentration as 10 milligram per ml, then in a given time what you are going to get, you are going to get the 10 milligrams of the B, right? So rate of reaction is always been dependent on the concentration of the reactants, right? And you can increase the rate of reaction simply by having a protein, protein which is going to cover these coiservates, and along apart from the proteins, you can also have the water molecule. So that's why it is actually going to form a small vessel like structure, and because of this the local concentration of the molecules are going to go up. So the membrane which is present around the molecule protect the molecule, right? So first of all it is going to protect, so it will going to be not going to be damaged, it is not going to disintegrate. So this is actually going to form the colloidal particles which are actually not going to dislodged, and at the same time it is actually going to bring the high concentration of the reactants to enhance the chemical reaction. So you can see when we were talking about the previous lecture, it took a couple of billion years for these reactions to complete, so that you can be able to have the synthesis of these biomolecules. But if you increase the reaction concentration, the chemical reaction are going to enhance, because remember that there is no enzyme, right? So there is no enzyme in this so the chemical reaction has to facilitate spontaneously on its own. The colloidal aggregates absorb the protein and the other biomolecule from the ocean. This results in the growth of coacervate as well as the internal complexity, as the coacervates divides into the multiple small ones. So now what happened is these coacervates started collecting the protein. So as an arrangement, the protein were present outside, and inside this structure they will be trapping the different types of biomolecules like the nucleotides or the DNA and RNA, and outside this you are going to have a water layer, and that is actually going to form a membrane like structures. And ultimately these coacervates started taking up the molecules from the outside, and that's how they will be started growing. So they will be started growing in size, and once they grow to a larger size, then these coacervates are breaking from the middle, and that's how they will be dividing into the two steps, into the multiple coacervates. And this is what is happening, right? So you have single coacervates. Around this you have the the protective membrane, right? This protective membrane is there, and then it actually grows in size. So if it grows in such a big size, then it divides and gives the multiple coacervates. And you can imagine that all these multiple coacervates again will do the same, right? They will grow, and then they will be actually going to divide, and that's how you are actually going to see large number of coacervates into the primordial oceans. Then we have the the two steps. Okay, so step is from the coacervates you are actually going to have the development of the protocell or the. So once the coacervates started taking up the biomolecules from the ocean, they have actually started developing the primitive cells. So coacervates are the initial species present in the ocean to start the formation of the primary cells. This process is accomplished in the two steps. So step 4 further been divided into the two steps. In the step 1 you have the formation of the protocells. The coacervates has the ability to take up the new molecule to replace the degraded molecule and maintain the size. So coacervates were the first species which are formed, and they were having the ability to replace the molecule which are being degraded, and that's how they can be able to do the if there is an injury, they can be able to do the repairing of that particular part. And thus the coacervates has the basic ability of the living system. It does not have the complex molecules such as the enzyme. The process of the acquiring new molecule was not regulated. Later, nucleic acid is entrapped within the coacervates, and the process of division becomes precise and controlled. This form of coacervate with nucleic acid is known as the protocells. Later on, what happened is the coacervates have started trapping the biomolecules. So initially all the reactions were very unregulated. So because they were governed simply by the mass balance, right? So wherever the mass is there, and if the mass is going beyond a size, it is actually getting weakened, and that's how it is actually going to break into the multiple portions. For example, this is what you see, right? This is a coacervate. It was very big, but at this point it got very high size of the biomolecule. So that's how this portion got constricted, and that's how this portion is going to be now form the new coacervates. So same is happening here also. So this coacervate is not having any regulation where this could be happening, but ultimately these coacervates have started taking up the the nucleotides or the nucleic acid. So once they have started taking the nucleic acid has taken up the because nucleic acid is having the information. So whether it is a nucleic acid of RNA or the DNA, that was having the information, and that they have started governing the activities within the coacervates, and that's how these coacervates which were having the entrapped nucleotides or nucleic acids are known as the eobuoyants or the protocells. So these were the first form of the living organisms. Later on, these protocells were form the first cell. So then the protein molecules and the appearance of enzyme has enhanced the synthesis of several of biomolecules which are present in the protocells. the RNA and DNA developed in these molecules, and that has taken over the protein synthesis. So initially the protein synthesis was happening because of the spontaneous reactions between the inorganic biomolecule or the smaller biomolecules, right? Because the smaller biomolecules were synthesizing the amino acids, and then these amino acids were binding to each other, and that's how the proteins are synthesizing. But as soon as the RNA and DNA were trapped within the coacervates, the RNA and DNA were guiding what will be the synthesis of the biomolecule. Although there are many things which are not known in this particular process, how the RNA and DNA were governing the synthesis of the biomolecules or the protein molecules and so on. So that these are some of the things which we are still not clear. The interaction of the lipid and the protein allowed the formation of the biomembrane, which has which has provided the selectivity in the primitive cell for intake or the exclusion. So ultimately what will happen is the people, the lipids are forming the aggregates with the proteins, and that's how they have formed the plasma membrane. So now there was a plasma membrane outside, and then you have the the nucleic acids in the middle, and that's how it has actually formed the first cell. And this first cell was primitive, and it was still need to be develop many things, like it has to develop the different types of organelles and so on, and that's how this primitive cell could be the prokaryotic in nature, and this prokaryotic cell could have given up the eukaryotic cells. It allows the appearance of the membrane bound protocell, and it has eventually give rise to the first cell on the earth. So this is what happened, probably could happen, and these are the proposed events. Still there are no experimental evidences to prove these points. These are the proposed events which are being proposed by the different scientists based on the experimental evidences, but not the direct experimental evidences through which the people are because we all still know that if you take the protein and lipids, and if you mix them or if you provide the energy into the system, they actually get rearranged to give you a plasma membrane. So these are the some of the experiment, but nobody has so far actually done these steps individually and to to create a protocell or to create the first cell. The mutation in the DNA and the selection of the fast growing give rise to the appearance of the first primordial cell. So ultimately there could be some mutation in the DNA, and that's how so the first cellular form was formed on the earth at approximately 2000 million years ago. So that's how the life is originated onto the earth. So this is all about the origin of life on the earth. If we summarize what we have just discussed, we have discussed the multiple events are required to form the life on the earth, and all these are the proposed events based on the results what people got from the Stanley Miller's and the Urey's experiments. So what he says is that the proposed events are actually a 4-step events. In the step 1, you are actually going to have the formation of the inorganic molecules, and then in the step 2 you are going to have the spontaneous formation of the simpler monomeric organic compounds. So in that you are going to form the monomeric amino acids and nucleotides, as well as the fatty acid and glycerol. And once you form the fatty acid glycerol, they are actually going to form and club together, and they were actually going to form the complex biomolecules or complex structures, complex biomolecules in the step 3. And then these biomolecules will come together, they will aggregate, and that's how they are actually going to form the coacervates in the step 4. And once the step 4, the coacervates are being formed, these coacervates are actually going to start acquiring the biomolecules from the primordial oceans, and they will be started growing in size, and that's how they will actually and apart from that, they will also have the ability to replace the biomolecules which are being damaged. And once they grow to a certain size, then these coacervates are actually going to divide, and they will give you the the daughter coacervates. But ultimately these coacervates have also started trapping the nucleic acids which are being also present in the primordial oceans, and that's how they have formed the first protocells. And these protocells, once they have having the nucleic acids, they have also directed the protein synthesis. The lipid also came into the picture, and the lipid along with the protein has formed the plasma membrane, and that's how you have the plasma membrane bound first cell which is formed within the primordial oceans. So this is all about the origin of life, and what we have discussed about the different theories in the previous lecture as well as in this lecture. And in this lecture we discuss about the modern theory of origin of life, and we have also discussed about the different experiments. So we have discussed in detail about the Steney-Miller's experiment and how the Steney-Miller's experiment prove that the smaller simpler biomolecules, similar chemical molecules can give rise to the complex biomolecules. One interesting thing which is very important is that people are still trying to discover the more and more molecules which the Steney-Miller has actually formed, right. So Steney-Miller has collected the different fractions, and then these fractions are still being preserved in his laboratory, and recently when the scientists have analyze those reagent bottles or analyze those fractions, what they could found is that because the Steney-Miller does not have the very high end technique in that particular time frame, he could not be able to identify some of the biomolecules. But now if you go and try to fractionate those molecules, what people have found is that even if they could be able to see the different types of nucleotides and other kinds of biomolecules which the Steney-Miller could not be able to report at that point. So this is all about the origin of life. In our subsequent lecture we are going to discuss about the evolution and how a simple cell which are being formed in the primordial ocean developed into the different types of organisms. So with this I would like to conclude my lecture here. Thank you.